Okay, looks like we are live. So welcome everybody to the next session. We just had a very nice session with all the data bits talks. It was very impressive. And I'm very happy to welcome you now on session 4, which is on aging and navigation. I don't know whether you can, how good my video is. In case it is good, please notice the shirt I put on, which has a nice hexagonal structure, just fitting for this meeting. Yeah, so we have 4 speakers tonight, or at least in Europe tonight, who will cover research, very exciting research, both with rodent models and with humans on aging, meaning how spatial computations change as we get older. And it's great to have a combination of, in the spirit of iNAV, to have a combination of behavioral and neuroscience work to bridge the gap across species. And we're going to kick off with Jennifer Bizon from the University of Florida, who talk about discrimination learning and spatial navigation in rat models of aging. So, Jen, the stage is yours.
All right. Thank you so much, Thomas, and for the invitation to participate. And I hope this is going to go okay. I've had a couple times getting kicked out here, so I'm hoping that this will be smooth. As Thomas mentioned, I'm Jen Bizon from the University of Florida, so it's only 1 o'clock in the afternoon here. And my lab focuses on really trying to understand brain aging and its contributions to cognitive decline in advanced ages. And I thought what I would do today in this talk is give you a brief overview of some of the features of rat models that we typically use in our research, as spatial navigation deficits are among those most well described. And then I'm going to review some of our relatively recent studies in which we've investigated perceptual discrimination deficits as a contributor to these spatial learning impairments. And then I'm going to spend the last few minutes showing you some of our unpublished and ongoing work in which we're introducing some pathological features of aging into this normal rat model with an AAV approach, and talk about some of the potential utility of this new model to reveal the influence of features of pathological aging, such as tauopathies, on functional outcomes relevant to spatial navigation. So I mentioned that we focus in rat models, and I've shown on this slide just some of the typical strains of rats that are used typically, at least in the US, to study normal aging and the age at which we consider these different animals aged. And we talk a lot within the field of differences across these different strains, but really, there are more similarities than differences. And some of the consistent features across all of these normal aging models is that we see generally a lack of a widespread neuronal loss in cortical structures. There's relative preservation of the neurons within the medial temporal lobe and cortical structures. We also see that these normal aged rats lack many disease-associated pathological features of aging, such as amyloid plaques and neurofibrillary tangles. And yet we see, as I mentioned, robust spatial navigation impairments. And this is what I'm showing you on the next slide. And this is the data from, these are, again, not genetically modified in any way, just young and aged animals. And with a standard Morse water maze task, we can detect robust spatial learning impairments in these aged animals. And these are data from my own lab over a number of years, pulled together across studies, but they're very indicative of what you see across all of these different rat strains. And that is that in the young and aged animals on the first day of training, these animals perform similarly. Both young and aged rats improve over the course of days of training on the water maze, but aged rats as a group are certainly impaired relative to young. Importantly, however, if you look at the individual performance of these animals, the aged rats tend to show greater individual variability than young, and that population actually overlaps with young. So some of those aged animals perform on par with their younger counterparts, whereas others perform outside that range of young and demonstrate varying degrees of impairment. Now, these types of spatial learning individual differences, or the individual differences in spatial learning deficits, have been used to try to dissociate general features of brain aging from those that are specifically relevant to spatial learning or cognitive decline. And I'll note here that although I'm not showing a lot of this data, we do know that these individual differences are not confounded by motoric impairments that can also associate with aging, and that these deficits don't necessarily indicate global cognitive decline, as we can also detect these types of individual differences on other types of cognitive tasks, such as working memory tasks that are, instead of hippocampal dependent, medial cortical dependent. So they seem to be very reliable individual differences that link very much to hippocampal deficits and spatial learning impairments. So what about the neurobiological mechanisms that underlie some of these spatial learning impairments? And certainly there are a number of things that have been discovered over the past 20 years or so, but one of the features that we've been particularly interested in relates to the idea that there is a shift in excitability within the hippocampus with age, and particularly within the CA3 region. And some of these data, certainly not exhaustive, are illustrated on this slide. And it includes things like the fact that there's interneuron loss from area CA3, as well as changes in neuronal activity detected with electrophysiology in aged rats within the CA3 region. And even in human neuroimaging, there is evidence of CA3 hyperexcitability. And this hyperexcitability in CA3 impacts, or we think that it might impact spatial learning because of the fact that, that there are these opposing processes within hippocampus that encode sensory stimuli. And one of these processes separates sensory information and encodes similar features as unique, and one pattern completes or generalizes across sensory information, and really consistent with the role for CA3 in pattern completion and this age-associated hyperexcitability change in this region. We really think that it's perhaps data or the changes in CA3 and their impact on the ability to discriminate similar features in the environment that may be at least one contributor to these spatial learning impairments. And so this is a question that we started asking a few years ago. As I mentioned here or showed here, that the behavioral deficits in aging that have really focused on discrimination learning of sensory stimuli have been mostly done in the visual domain. Rats are, in fact, olfactory creatures, and they tend to rely most heavily on olfactory cues to navigate their environment. And in fact, the perirhinal and entorhinal cortices that provide hippocampus with this highly processed sensory information that's so important for encoding new memories, the olfactory information within rodents in that system is quite robust. And in fact, what I'm showing on the right here is that young and aged rats can detect and respond to odors very comparably. So they don't have any problem detecting odors, suggesting that olfactory cues may be very well suited for examining sensory discrimination. And in fact, olfactory cues are well suited for grading perceptual similarity, which is what I'm going to show you on this next slide. In many ways, the olfactory domain lends itself to asking questions about perceptual discrimination abilities, as olfactory stimuli can be very easily varied parametrically. And specifically, within a chemical class such as the straight chain alcohols that are shown here, you can vary the perceptual similarity of the odorants by varying the number of carbon atoms in the carbon chain backbone. And this example shows that, for example, propanol and octanol can differ or differs by 5 carbon atoms, and that's more perceptually distinct than would be butanol versus heptanol, which only varies by 3, and that will be more distinct than pentanol versus hexanol, which only varies by 1 carbon atom. And in work done by Wendy Yoder in the lab, this first graph shows that in young animals, if you train animals in a go, no-go olfactory discrimination task, that you see this nice gradient of accuracy depending on whether the odorants presented differ in the number of carbon atoms, as I just described. So the work I'll show you next is a continuation of Wendy's work, and now in the context of aging. In this first graph, you can see that if we look at young and aged performance on olfactory discrimination problems using stimuli that are structurally unrelated, young and aged rats perform very comparably. However, if we look across a class at those different problems that differ with respect to perceptual similarity, you can see that the aged rats in orange are actually disproportionately impaired relative to the young. So although that both young and aged show less accuracy as the problems become more perceptually similar, the aged rats show a more pronounced deficit. So the next slide, I just want to make the point that we don't think these discrimination deficits are selective to the olfactory domain. And in fact, in very nice work done by Dr. Sarah Johnson and Dr. Sarah Burke, they have done essentially comparable experiments using visual cues in rodents where they varied the sensory overlap of the stimuli using Legos. And you can see that their young and age data from these visual discrimination problems look very similar or comparable to that we see with the olfactory discrimination. So what about in relation to spatial learning? This is now showing the same type of data, but now I've collapsed it across all of the trials within a session. And these animals had been actually cross-characterized on spatial learning task as well as the sensory discrimination task. And here you can see that the young animals and the aged rats that were spatially unimpaired or performed like young on the water maze task actually performed very comparably on these discrimination problems. And it's really the aged rats that are spatially impaired on the water maze that show this decrement in accuracy as these discrimination problems become more perceptually similar. And that's also illustrated with this correlation among the age subjects. So,
Jen,
yes.
Sorry, Jen, we have about 4 minutes left.
Okay, perfect. That's perfect. And so that's actually and so what I'm going to talk about just for these last few minutes are a couple of the directions that we're moving in now. And I was really hoping to have new data for this part to present in Italy in June. But of course, with COVID, we had to curb a lot of experiments, as did most people. And so we're getting these back up and going. But I do want to mention two of the directions that we're going now. And one of those questions, now that we have established this relationship, is to try to understand how these perceptually similar and distinct pairs are being encoded within the medial temporal lobe in aged animals that are spatially unimpaired and spatially impaired. And again, this is work in collaboration with Drs. Burke and Johnson. And this just shows immediate early gene expression in a young animal that has been trained on this task and that we can detect very robust immediate early gene expression throughout all of these regions. And so now we're in the process of doing this in spatially characterized young and aged animals. And then the second direction that we're working on, and I'll just mention this very briefly, is to try to understand. I mentioned that these normal aged rats that we have been studying for many years don't exhibit a lot of the AD type pathology like neurofibrillary tangles, but we know that tau pathology is happening both in normal aging as well as, of course, in AD. And as everyone probably knows, both discrimination learning and spatial navigation are being explored as potential predictors of AD. And so we were really interested in whether we can take advantage of this robust cognitive context that we have with these aged rats to try to introduce AD-like pathology, particularly tauopathy, in these animals to start to understand the interactions between aging and tauopathy. And while I don't have a lot of data on the behavioral side to show you, I will mention that we've made considerable progress here. This slide just shows, this is the work of Dr. Joe McQuail in the lab, that the young and aged rats' endogenous tau doesn't change as a function of age in this model, and nor does phosphorylated tau. And this is from biochemical studies from the medial temporal lobe in young and aged animals. And so against that context, we're now using an AAV approach to inject humanized wild-type tau into the medial temporal lobe. And these data, which I won't go into detail, are just showing that we can robustly, 2 months after we inject the tau construct, see pronounced hyperphosphorylation of tau in both young and aged animals. And this shows that this tau pathology is actually potentially, and this is ongoing, more pronounced in the aged animals than our young subjects that received this construct. And then finally, I'll just point out that in this, and then we've also examined this within respect to all of that work has been done in the perirhinal cortex where we're injecting the humanized tau. But in collaboration with Peter Rapp, we've also done some iDisco in these animals 2 months after we've injected and done parallel biochemistry experiments to show that we also see quite a considerable phosphorylated tau in the hippocampus of these animals when we're injecting them in the perirhinal cortex. And so I won't go through all the summaries, but the next thing is, of course, to try to understand how this tau pathology influences discrimination learning and spatial navigation in the aged rats. So I mentioned a lot of my collaborators as we go, so I won't take time here, and I'll leave some time for questions.
Okay, great. Thank you very much, Jen. Very interesting talk. Okay, so I'm reading to you the question. So the question that received most of the votes is from Kevin Allen, who said, great work. When stimuli are more similar, older animals are impaired. Does that necessarily mean a problem for older animals with pattern separation? Could there be, for example, deficits in inhibiting responses to more ambiguous stimuli?
Sure. I mean, that's a great question. And it could be. I'm trying to think. It certainly could be because it is a go, no go task. I will tell you that we've also done this type of work in a task in which we have 2 choices. It's a go task, and we see very similar types of deficits. So I don't think overall that it's an inability to withhold a response, for example, but we haven't necessarily done those 2 things in conjunction with the sensory ambiguity. So that's a really good question. It'd be a great thing to follow up.
Okay. Next question. Are the animals also impaired in a non-spatial water maze task, so with a cube platform, just to know if it's that they don't see well or it's related to memory?
Sure. So none of these rats are impaired in a cued version of the task. So we've very exhaustively evaluated all types of motoric impairments, cue training, and other types of general sensory motor dysfunction, which you can certainly detect in older animals, but these deficits, to the best of our ability, are not due to those types of impairments.
Okay. And maybe one last question. So have you tested if the deficits in discriminating perceptually similar odors can be explained by deficits in the olfactory system?
Yeah, so there may be, and so this is something that we, there's some work certainly that suggests that changes in the olfactory system, particularly there's some nice work on cholinergic signaling within the olfactory system that's been linked to this type of discrimination deficits. And I wouldn't at all be surprised if that's a contributing feature. I think, you know, the combination of the work that Sarah Burke and Sarah Johnson and our labs have been doing in collaboration to us suggests that these things, that this discrimination is maybe not specific to or restricted to olfaction, but certainly changes in the olfactory system could be contributing to it as well.
Right. Okay, and if I may sneak in one question myself.
Sure.
You mentioned like, for example, in early Alzheimer's, like emerging tau pathology in transentorhinal cortex. So there was a paper, I think by June et al. In Neuron a couple of weeks ago, who basically said, okay, we see a breakdown of the grid code, and that could actually explain or lead to pattern separation deficits in hippocampal place cells. I think it was the CA3. Have you thought of, do you have any data? Have you thought about the role of entorhinal cortex in actually producing these pattern separation deficits? So is the pattern separation inherited from maybe entorhinal cortex, or what is your take on that?
Right. So we actually are focusing almost more on perirhinal, entorhinal than we are on CA3, to be honest. And that's really driven by Sarah Burke, who's my collaborator, and that's, mostly her line of work is focused on that area. But that's where a lot of our future work is going. But we're interested in the connections. So we're trying so far with this tau model, we've been doing a lot of biochemical and just trying to understand what we're seeing 2 months and 6 months out after we do the AV injection. We have very lofty plans in the future of trying to record from these areas. And by we, that's the royal we, meaning Sarah and her group. And then also really characterize some of the different cognitive changes that we know we can dissociate perirhinal from hippocampus, for example.
Okay, great. Okay, well, I think we have to move on. So thanks very much for this great talk. I hope you enjoyed the format and also the discussion. And,
Thank you.
And we're going to move on to Jennifer Campos now, already here on stage. Jennifer, can you hear me?
I can. There, I'm unmuted. Okay, perfect. Okay.
Well, we can hear you too. Great, fantastic. So our next speaker is Jennifer Campos from University of Toronto. And she's going to talk about multisensory self-motion perception in older and younger adults. So Jennifer, just share your screen, and I think then we should be good to go. There we go.
Okay, you can see that fine. Great. Well, thank you so much for the invitation to present today. I'm joining from actually sunny Toronto, and I'm going to tell you a little bit about our work on multisensory self-motion perception in younger and older adults. So many of you are probably not familiar with the Toronto Rehabilitation Institute, but it is the largest adult rehab hospital, teaching and research hospital in Canada. It's very interdisciplinary, lots of computer scientists and engineers working alongside clinical scientists and fundamental basic science researchers. And the focus is really a lot on translational research goals, including prevention of injury and illness, restoration of function following injury or illness, and the promotion of aging well. And so in order to kind of meet some of these goals, we do use in some of our research a collection of virtual reality and simulation labs. So what you see here is sort of 4 module labs. We've got Street Lab here, which is our immersive virtual reality simulator. We have Driver Lab, our driving simulator. We have Winter Lab, which is our climate controlled space, and Stair Lab, where we can look at various features of the built environment. And these are all actually portable labs, and they can be placed on top of this large motion platform. And this is 6 degrees of freedom, so it can move in X, Y, Z dimensions and rotate and yaw, pitch, and roll. And so the advantage of using these simulators, of course, is that we can start to study some of our research questions in more controlled but realistic and safe settings. But from the perspective of studying multisensory self-motion, which is my main interest, is that one of the key attributes of VR and simulators is that we can carefully control the sensory channels. We can manipulate them, and we can control the availability of different sensory cues and look at the effects on perception and behavior. You'll notice I put some QR codes also throughout my presentation in case you want to capture those for more information. But as I said, my research interests are largely in the area of multisensory self-motion perception across the sort of adult spectrum. And this includes understanding how we integrate information from our vestibular system, visual, auditory, and proprioceptive systems to help control standing balance, to estimate parameters of passive self-motion, and also to understand active self-motion or perceived active self-motion like walking and driving. But of course, our sensory systems are not always 100% reliable. We have noise in our sensory estimates, and we have environmental challenges that we're often met with. So it really benefits the brain to be able to take advantage of these congruent, redundant sensory signals. And usually we see under multisensory conditions that we have perceptual and behavioral benefits from integrating across these signals. And we also tend to see that there's a reliability-based weighting of these different signals, so that the more reliable signal in any given situation is weighted higher than others. But of course, in addition to these kind of environmental challenges, with age, we often experience declines across our sensory systems, and that can also change the way in which we integrate across sensory inputs. So when we think about the ability to estimate different parameters of self-motion, when you're young and you have highly reliable sensory signals, we can actually do a fairly good job at using one or other of those signals to be able to estimate these parameters alone in many cases. But of course, there's always going to be some benefit, or often going to be some benefit, combining across the senses. But when we consider what happens when we get older and we experience age-related kind of normative declines across our sensory systems, what happens is it actually becomes even more beneficial for us to take advantage of multiple redundant cues than it does when we're younger. And so our group and many groups before us have showed that in many situations, older adults demonstrate evidence of heightened multisensory integration. And if we consider cases of like clinically significant sensory loss, then of course this changes also the way in which we weight the different sensory inputs. And so these general principles of multisensory integration in terms of multimodal benefits, especially in the case of weak inputs, we see this at the level of single multisensory neurons and firing rates and single multisensory neurons in the brain. And it's also been demonstrated quite well for very simple behaviors and basic tasks, but there's really not much that's known about how these principles apply to more complex tasks, especially in the context of self-motion perception. And so I'll start off by telling you a little bit about a very simple study that we did in collaboration with our colleagues at Toronto Rehab. It was led by my former PhD student, Robert Ramkalawansing. And the participants, I'm going to tell you about 3 studies. Across all studies, unless otherwise stated, we tested both younger and older adults. And when we talk about older adults, they're 65 years and older, and they were pre-screened using standard metrics to assume that they do not have clinically significant sensory loss, even though they may be experiencing typical age-related decline. So that they had normal sensory, motor, and cognitive abilities. So for this first study where we looked at driving in older and younger adults, the task was really very simple. So they were in this virtual environment and they had to keep their speed, maintain their speed, and stay within their lane. And we had 3 different conditions. One in which there was only visual information to help them guide their speed maintenance and lane keeping. A condition where they had auditory feedback from tire roar, wind sounds, and engine noises. And then a condition where they had the availability of motion that came from our 6 degree of freedom motion simulator. We looked at a lot of different metrics, but I'll just briefly tell you about what we found with respect to speed variability, mean speed, and variability in lane keeping. So what I'm showing you here is the results for speed variability, and you'll see I've plotted the younger adults on the left and the older adults on the right. And what might be immediately apparent is that for younger adults, we do see some benefit for adding auditory cues. They reduce the variability in their speed keeping, not so much for motion. But what's really interesting is for the older adults, we see a much bigger difference. And so when you add additional auditory feedback and motion feedback, older adults reduce the variability in their speed keeping quite significantly. And we also saw some evidence of this for the mean speed, where older adults slowed down when auditory cues were added. A little bit of a different story when we look at the variability in lane keeping. So again, younger adults on the left and older on the right. You can see that for younger adults, if we added auditory or motion cues, it didn't really change their ability to, for lane keeping. Obviously, we wouldn't really expect much from auditory inputs in this case. But when you look at the older adults, they actually showed an interesting increase in variability in their lane keeping when motion was added. So it was a bit of a curiosity at first, but of course, when you are using a motion simulator, you're using motion cueing algorithms, which rely on a little bit of trickery and don't have the full motion capabilities that you would normally have in the real world. So we were wondering if perhaps it was the case that older adults were sensitive to some of these subtle incongruencies that were introduced, but were still attempting to integrate those signals, and that may have led to more variability in their lane keeping. But of course, there's a bit of a confound there, because we'd want to know, is it really due to greater sensitivities in sensory conflict or multisensory explanations? But confounded with age is also cumulative driving experience. So older adults do tend to have more experience driving, and so they may have more kind of sort of robust learned associations between what to expect from the visual motion and the physical motion that they would be getting. And so what we did is we actually ran a very stripped down version, and we did a psychophysical heading discrimination task. So in this case, we put people on the motion platform, and we moved them, we translated them forward to the left or to the right, and we gave them two motions. So they would do one motion and they would be passively moved in another direction. And their task was to judge in which of those two motions did you move more to the right. And so we did this across 3 different sensory conditions. The first one was visual only. They saw through a head-mounted display a basic optic flow cloud of dots. Vestibular input alone, so passive motion in the dark, moving along that same raised cosine velocity profile, or combined inputs. So they had both visual information and the motion inputs combined at the same time. And so here, summarizing again, we had visual alone, vestibular alone, and combined and congruent. And we presented them at various different movement angles. And what we're interested in is their precision with which they're able to differentiate these different movement angles. And what we expected to see is that if you have combined visual plus vestibular information, then you would have a greater precision in your estimates compared to if you had just the unisensory information alone. But we also added, in addition to the congruent combined condition, we also added and introduced a sensory conflict. So in this case, what we did for these conflict conditions is that in a small conflict condition of the 5 degrees, physically they would still be moving straight ahead, but visually we'd actually be presenting them at the heading angle of 5 degrees to the right. And for the large discrepancy, the large conflict, the same thing was true where physically they were moving straight ahead at 0, but visually they would be moving at 20 degrees to the right. And so what did we find? So when we look at just the vestibular alone condition, both older and younger adults estimated heading equally well with vestibular cues alone. There was actually no difference in their ability to do that. When we look at the visual cues alone, estimates were less precise for both groups, but older adults were even less precise than younger adults when having to use visual information alone. When they were combined and congruent, both older and younger adults integrated visual and vestibular cues optimally, as we had expected. But most interestingly is when we had them combined, but in conflict. And so when we had those 5 and 20 degree conflicts, it did cause optimal integration to break down. But this breakdown actually happened at smaller conflicts, the 5 degree conflicts for older adults compared to younger adults. And because we can look at sensory weighting, because we presented in the conflict trials a heading direction that was different visually than it was physically, we also observed that while younger adults always weighted the more reliable input higher, in older adults, vision was actually given more weight than would have been optimal, remembering that vision was actually the less reliable signal in this case for both groups.
Yeah. Jennifer, you've got about 3 to 4 minutes left.
Perfect. So I'll just finish off by telling you a little bit about some of the work we're looking at hearing loss and self-motion. I was very happy to hear the auditory system mentioned a few times this morning, but it does seem a little bit surprising in a navigation conference. It's not something that's often considered in humans. And so there are many good reasons to consider hearing loss. One is that it's, especially if you're doing aging research, it's a really highly prevalent chronic health condition. In Canada, it's the 3rd most prevalent chronic health condition, and it affects about 50% of people over 70 years of age. But something I'm going to focus on today is that it's being there's emerging and convincing evidence that individuals with hearing loss are at greater risk of mobility problems, and some work by Frank Lin and his colleagues in a large population study showed that individuals with even mild hearing loss are at 3 times greater risk of falls, and that this increases with the increased with increased hearing loss, you have increased falls risk. The thing is, we don't actually know why. We don't know what that association is, and there are some probably non-exclusive or exhaustive hypotheses that I've indicated here. But I'm just going to tell you about this one today, which is that there may be paralleled vestibular loss in age-related vestibular loss that goes along in some cases with age-related hearing loss. They sort of work in similar ways. They're in close spatial proximity, and they have shared nerve inputs to the brain. So it seems reasonable. This is a collaborative effort, and it was led by Grace Gabriel, a PhD student in my lab. And so here in this case, we did have the younger adults and older adults, but we also included older adults with age-related hearing loss. And we wanted to make sure they didn't have any clinically significant vestibular loss, so we did additional baseline tests to rule that out. And in this case, we used an adaptive staircase procedure, a passive self-motion perception task, where we move people in heave and pitch. And we had them do 2 tasks. In the movement discrimination task, they were presented with 2 heave motions or 2 pitch motions, and they were asked to determine in which of those 2 presented intervals did you move more. And then a movement detection task. So they were presented with 2 intervals, and they were asked simply in which of those 2 presented intervals did you move. And so I'm not going to show you the results of heave because we didn't actually see any differences as a function of age or hearing status in heave. But pitch detection, you can see that the older adults had higher thresholds than younger, so we see an effect of age. But when we look at pitch discrimination, we actually do see a significant increase in pitch discrimination thresholds in individuals, older adults with hearing loss compared to those with normal hearing or younger. So to summarize overall, in our driving study, we showed that older adults showed greater multisensory effects during driving than did younger adults. And in some cases, those are beneficial, but in the case of maybe some conflict or discrepant cues, it could be detrimental. In our heading discrimination psychophysical task, we saw that older adults showed evidence of optimal multisensory integration, but lower tolerance to sensory conflicts, and this higher than optimal weighting of the less reliable sensory input. And for hearing loss, we saw some evidence that individuals with age-related hearing loss may also have less sensitive vestibular percepts. So it's an argument, I think, hopefully you've been convinced that considering multisensory contributions to self-motion perception is important, and that there may be age-related differences in implications for potentially some applied areas. And I'd like to thank my team.
Perfect. Thank you very much, Jennifer. Great talk. So we have a couple of questions. Let's just move straight in. First one, does heightened multisensory integration with aging also mean that older individuals are more sensitive to conflict between sensory modalities?
Yes, so I think that's what our research shows, and others as well. So heightened integration can be positive. Obviously, if it's redundant and congruent, it can benefit percepts. But if there are some discrepancies between sensory inputs, if they are kind of widely spaced in space or time, that sort of the temporal and spatial binding windows we call are a little bit older larger in older adults, that could also be detrimental if it's not related to the same event or object. So in general, I think in a world where things mostly are congruent and consistent and redundant, it can be beneficial, but not always.
Right. So next question is about the type of visual stimuli. So great talk. Do you think the overweighting of vision in older adults is specific to the stimuli presented, or would it be the same in a realistic setting like the driving task?
Yeah, so you could see both in the driving task, which did have some, monocular depth cues and some contextual information, that the older adults did worse in the speed, like they had higher speed variability, even in the context of those visuals. When you strip it right down to an optic flow display, then they also showed worse performance than younger adults. But the interesting thing about that was that even though they had less reliable estimates for those types of visual stimuli in both of those cases, at least in the psychophysical task, we still saw that they weighted those unreliable visual cues higher than they should have in the combined estimate. And so that, it most certainly has to do, I think, with just in general, older and younger, the extent to which you get really reliable and robust information from vision is going to affect its relative weighting. But it is interesting to see that older adults don't necessarily optimize it.
Okay, if I may ask a question related to that. So in some of our work, we also see that in particular, when you add a memory component, when they have to navigate, say, based on self-motion, but then you also add a memory component, then the deficits become really striking for self-motion conditions. Have you looked into memory at all, or are you just more focused on the perceptual integration here?
No, we have looked not as much at memory, but definitely at introducing cognitive load. So we will introduce cognitive load in the context of dual tasking, for instance. So they'll have to perform, for example, a listening task and have to walk or balance at the same time. And absolutely, we definitely see much bigger age-related effects as soon as we add a cognitive load component. And that's also one of the kind of suggested mechanisms or reasons underlying why people with hearing loss are at increased falls risk, is that when you have hearing loss or a sensory loss in probably in any domain, that it increases the load in having to, even just the listening load, the ability to listen, and it could tax cognitive resources, taking those resources away from the other things that you're trying to do, like supporting mobility. And so we've seen work in our lab where in addition to the healthy older adults, where there's bigger constraints that are placed and bigger effects due to these cognitive demands, we see that with individuals, older adults with sensory loss, those differences are magnified even more.
Okay, let's wrap it up with two quick questions. First of all, a more methodological question. How big of a problem was motion sickness in the sensory conflict study, and were there differences in older adults? And a second question from Hugo Spears. Do you have any insights into how superagers might perform?
Okay. So in terms of the motion sickness, in the psychophysical task, we had very little motion sickness. So when you have these really basic optic flow stimuli, we don't typically see very high rates of simulator sickness, including in older or younger. In the driving task, we did see higher rates of simulator sickness, and there is a precedence in the literature that might suggest that older adults would have higher rates than younger adults. And we saw a little bit of that, but it really wasn't sort of modulated by sensory condition, which was interesting. In terms of super performers, I would actually argue that most of the people in our studies are super performers, or kind of cream of the crop older adults, because we do that very intensive battery of baseline tests to look at their sensory, motor, and cognitive functioning at baseline. And we use that as an eligibility criteria for most of our studies. So we're really interested in kind of healthy aging. And so people who have any kind of neurological or major sensory impairments that we're not kind of focused on, motor impairments, etc., cognitive impairments, they wouldn't actually be part of our sample. So we'd like to actually do this work with a much broader range because we're seeing it even in these sort of high performers. So it's probably underestimating what's going on in the general population.
Fantastic. Okay, great. Thank you so much. Very interesting. We could go on for a long time, but now we have to move on to Jan Wiener. So thanks, Jennifer. Have a good day in Toronto. So we're leaving now North America and move to the UK to Jan Wiener. And Jan, can you hear me?
Yeah, can hear you well. Can you hear me?
Yes, fantastic. So do you want to bring on your slides? So next speaker is Jan Wiener from Bournemouth University, who's going to talk about from repeating routes to planning novel routes, the impact of landmarks and aging on route integration and cognitive mapping.
Okay, thank you very much for inviting me, Thomas. And
I don't think we see your slides yet. Can you move there?
Okay, hang on. That's better?
So maybe can the audience tell me? So I don't see any slides. Does anyone to the audience? Do you see slides from Jan? No.
No?
No. Looks like I don't see slides.
That worked yesterday.
Are you? Maybe someone from the backstage crew could help us.
Hi, Ian. This is Tamara. Can you hear me?
Yeah, can you?
Yes. So to share your screen, hover your mouse on top of your video, and on the top there should be an icon that is a laptop with an arrow. If you hit that, it will. There you go. So there you go.
There we go. Okay, great. Okay.
Well, thanks again for inviting me, Thomas, and the organizers, and thanks to everyone who makes the iNAV happening. So I want to give you a little bit of context. I'm going to present one study that is part of a larger project that's called Dementia-Friendly Architecture. And really in the project, what we're trying to do is we're trying to understand how both typical and atypical aging affect navigation behavior and navigation strategies, and how we can design environments to support navigation and to compensate for declining navigation ability. So that may explain why our virtual environment looks a little bit like a residential development or a care home. So this particular study that I'm going to present in a bit more detail is really about how we integrate routes that we have learned to form a cognitive map, and how landmarks and cognitive aging might affect this process. So the integration of overlapping routes has long been considered a crucial process for cognitive mapping. And for the purpose of this talk, I'm going to define cognitive maps as any representation of space that supports goal-dependent, flexible navigation behavior. Now, route knowledge is typically conceptualized as stimulus-response associations, and these obviously do not allow for flexible navigation behavior. So these somehow need to come from stimulus-response associations to a representation that allows for flexible navigation behavior. And more recently, several studies suggested that route knowledge is best described as stimulus-response-stimulus associations. And of course, stimulus-response-stimulus associations, they allow anticipating upcoming places given particular movement decisions. And this ability to anticipate upcoming places given a particular movement decision is a prerequisite to behavioral flexibility, as it allows comparing outcomes of different actions and selecting between them. So if independent overlapping routes that are represent a stimulus-response-stimulus associations are merged into an integrated representation of space, true goal-dependent flexible navigation behavior can be achieved. And let's look at this in a little bit more detail. So this is is an environment that looks very similar to the environment we use in our experiment. So assume you learn two routes from A to B and a route from C to D. The first thing in order to integrate those routes is that you need to understand that these different routes are traversing through the same place. Once you understand this, you need to have an understanding of the relationship between the intersections and the different paths. In order to, at the end, come to, this is just an overlaid representation of both of these routes. Now, if each place can become a start place and a destination, then you have a truly integrated representation of space that allows for both different flexible navigation behavior. we believe that the difficulty or the nature of this integration process depends on the information that is available at the intersection. So if the intersection has a similar visual appearance from all directions, integration really requires an understanding of the exact spatial relationship, that is the configuration of these different route segments. However, if the intersection contains distinct landmarks, these can be associated with different approach directions to either support integration or to allow for a goal-dependent flexible navigation behavior with a representation that doesn't require configural knowledge. So our first question here is, how does landmark information affect route integration? Let me give you a little bit of background on cognitive aging. We know that cognitive aging is now associated with performance declines in both route learning as well as in cognitive mapping or cognitive map usage. Route learning is associated with the use of egocentric reference frame. Imagine turn left at gas station or turn right at the street lights. The actual direction information is encoded in an egocentric reference frame. However, cognitive mapping or cognitive map use is typically associated with allocentric reference frames. We also know that cognitive aging is associated with larger difficulties in tasks that cannot be solved using egocentric strategies alone, and this really suggests that cognitive aging may specifically affect the process of integrating route knowledge. And one question is whether strategically placed landmarks can ameliorate potential age-related integration difficulties. So to answer these questions, we designed two virtual environments that look very similar to the schematic drawing I've just shown you. So we have 4 different rooms, and they are connected by corridors, and there's a central intersection. And in the different landmark environment on the left-hand side, we place 4 different landmarks, 2 of each type, at the beginning of these corridors. And that then results in intersections that look different depending on the approach direction. In the identical landmark environment, on the other hand, we use the exact same landmark and we present it in the same positions as in the different landmark environment. And now the intersections look identical irrespective of the approach direction. So note that the rooms cannot be seen or distinguished while you're navigating through the environment, so while you're navigating towards the intersection. So the procedure of the experiment is as follows. The experiment consists of several experimental sessions, and each experimental session comprises a learning phase and a test phase. And during the learning phase, participants are passively transported along 2 routes from A to B and from C to room D, and we use the same routes in each experimental session. The test phase then consists of 12 navigation tasks, and importantly, participants don't receive any feedback during the test phase. So they're navigated to the intersection and they're asked, in order to continue along the route, do you need to go left, right, or straight? Once they made their response, then they enter the next task. So during the test phase, the 12 routes can consist of 2 tasks in which participants have to repeat the learning route. So learning routes from A to B, and they are placed at A. They're asked, your task is to navigate to B. You navigate towards the intersection, have to decide left, right, straight. We have 2 tasks in which participants have to retrace the learning routes. That is, they start from B and then have to navigate to A, or they start at D and then have to navigate to C. And then we have 8 novel route tasks, and these are all of the remaining combinations of start, places, and destinations. So A to D, A to C, B to C, B to D, and so on and so forth. Now, importantly, the novel routes allow us to probe integrated route knowledge. So you can only navigate the novel routes if you have integrated the knowledge that you have learned when learning the route A to B and the route C to D. The participant has to go through 8 experimental sessions, or until they reach a performance criterion of 90%. 90% means they have to get 11 out of the 12 test trials correct. And by that time, if they reach the criterion, we assume that they have learned the environment and they show goal-dependent flexible navigation behavior. The DVs, the dependent variables we're interested in, are the number of sessions participants need to reach the criterion and the performance for the different route types. So let me get to the results. Oh no, sorry, this is just an example of a test trial. So participants would see a screen like this. The task is to go from the garden room to the cinema, and after this would be on the screen for a few seconds, and then they would see a camera movement from the garden room towards the central intersection. At the central intersection, movement stops, and they're given 3 options, either to go left, to go straight, or to go right. And once they've made that decision, the next test trial would start. So no feedback. We had 119 participants. 52 of our participants were younger adults, aged 22 on average, and 67 older adults, aged just under 71 years. We assessed cognitive functioning using the Montreal Cognitive Assessment. And for those of you who are not familiar with this, this is a 30-point screening tool for mild cognitive impairments. And there are different suggested cutoffs. We use the more conservative cutoff of 26 here. So 26 points, if you score 26 or above, you consider to age normally or healthily. This was a between subject design, so participants either took part in the different landmark condition or in the identical landmark condition. Okay, let me get to the results. First, what you see here on the left-hand side is for all of those participants who have reached the learning criterion, the number of sessions they needed to reach the criterion. We have our older participants on the left-hand side and our younger participants on the right-hand side. And you see there is a clear effect of age. Our older participants needed more sessions to learn the environment than our younger participants. There's also an effect of environment. So we do find that the different landmark environment in light gray bars was easier to learn than the identical landmark environment in dark gray bars. However, there was no interaction. That was something we would have expected. This is just a cumulative plot that demonstrates the percentage of participants who have reached criterion over the experimental sessions. Now, I said this is an analysis for all of those participants who have reached the learning criterion. There were a number of participants who didn't reach the learning criterion. Only one of our younger adults didn't reach the learning criterion. 37, that is 55% of our older adults reached the criterion, and 45% of our older adults did not reach the learning criterion or did not complete the experiment. So quite a few participants said they wouldn't want to continue with the experiment. And while we haven't formally assessed this, they suggested that the experiment was just too difficult and they didn't really want to continue. So we do find age difference despite essentially screening for good navigators in this particular analysis. Now we can look at the different route types, and this is an analysis in which we look at the novel routes, the retracing routes, and the repetition routes in the 2 environments. And here we don't find an effect of environment. However, we do find an effect of route type. Novel routes were the hardest routes. They were more difficult to navigate than retrace and repetition routes, which didn't really differ. And there was a significant interaction between route type and environment, and that interaction shows that planning novel routes was particularly difficult in the identical landmark environment, and that is the environment that requires knowledge about the exact spatial configuration of groups in the environment. Landmarks in the different landmark environments seem to have facilitated route integration. We can also look at the different participant groups and their performance in the different route types. On the left-hand side, you have our O- participants. Those are the participants, our older participants who didn't reach criterion, our older participants who reached criterion, our young participants who reached criterion. And we do find performance difference between all 3 groups. But importantly, we do find significant interaction between route type and group. That was driven by particular low performance in our older participants who didn't reach criterion when they had to retrace routes or when they had to plan novel routes. So this really shows that our O- group had particular difficulties with navigation tasks that could not be solved with relatively simple egocentric navigation strategies. So,
Jan, we have about 3 to 4 minutes left.
All right. So to further look into the difference between the different routes, we looked at their Moca scores, which assess cognitive abilities, and those different between participants groups. Specifically, our older participants who didn't reach criterion, this is essentially a histogram here, they showed lower performance on the Moca than our older participants who reached criterion, which showed a similar Moca score than our young participants. And in order to assess the relative contribution of age and Moca or cognitive abilities, we ran a multiple linear regression, and we found that a Moca score was a significant predictor of performance. However, age did not predict performance at all, and there was no interaction. So our O- group, the participants who didn't reach criterion, scored on average 25, which is just below the suggested cutoff of 26 that indicates healthy aging. And it really demonstrates that they have showed earliest signs of atypical aging, and it is that atypical aging, not age itself, that predicts spatial learning and navigation performance or navigation deficits in our task. So let me discuss this. We do find faster learning and better navigation performance in the different landmark than the identical landmark environment. And that just goes to show that landmarks at intersections do support spatial learning and navigation. However, differences between the two different environments, the different landmark and the identical landmark environment, were driven by novel route trials, which require route integration. So this shows that landmarks specifically supported route integration, and in the different landmark environment, participants can use the associations between landmarks and routes to plan novel routes. Potentially, they don't even need to have a configural knowledge of the environment. In the identical landmark environment, in contrast, participants need to have configural knowledge of the environment. So that shows that strategically placed landmarks can support route integration and goal-dependent flexible navigation behavior, potentially without configural knowledge. And second last slide, we do find that our younger participants showed better learning and navigation performance than our older participants, and this is in line with the reported age-related declines at forming spatial representations. We do find that almost 45% of our older adults never learned the environment, and those are the participants who performed low in the screening tool for mild cognitive impairments, and I think we have to assume that they show earliest signs of atypical aging. Importantly, this group performed at chance level for retrace and novel route trials, and those are the trials that cannot be solved with egocentric navigation strategies alone, and just confirms that atypical aging is associated with difficulties in non-egocentric navigation, route integration, and cognitive mapping. Finally, navigation performance was not associated with age itself, but with cognitive abilities or declines in cognitive abilities, and this highlights the differential impact of aging with typical and atypical trajectories for navigation. But it also, for us doing aging research, suggests that we need to carefully consider what kind of screening tools we use and what kind of cutoffs in these screening tools we use in order to avoid overestimating the effects of healthy aging. And with that, I would like to thank you for your attention and thank my collaborators and co-authors, particularly Ramona Kreshik, who ran this study when she was a postdoc in my lab. Thank you very much.
Great. Thank you very much, Jan. Very, very interesting talk. So, get the question here. There's a question from Kate Jeffrey that I think received most of the votes. Really nice effects. To what extent do you think these results could be explained by a failure of establishment of a stable head direction signal in the older participants?
I'm not entirely sure how important the head direction signal is. Participants are sitting in front of the screen and they move towards a single intersection. They see a single left turn or a right turn. So I'm not entirely sure how precise you need to know your orientation in space. So this seems to be all very left turn, right turn, or straight. This is all the information that you need, I would assume.
Do you have, maybe just to add to that, do you have any other data where there's more continuous movement, say more continuous turns, where you could look at that?
We don't currently have any. We have Chris Hilton, who is a PhD student just finishing up his PhD. He has recently done a study in which he looked at learning performance in a more naturalistic environment and then asked participants to point towards different objects or landmarks along the route. And there he finds aging effects, but he also finds that sequence knowledge, which goes back to our stimulus response stimulus knowledge of route knowledge, seems to be very predictive for performance in pointing tasks, but direct evidence we don't have.
Okay, great. So next one is by Arne Ekstrom. Nice talk. Given that older adults struggled the most with visually identical routes, would the addition of vestibular cues potentially have changed their performance?
I believe that, it's a similar response to the head direction self question, I guess. if you navigate through this environment, it seems to be so clear that you're doing a left turn or a right turn, and this is the simplest, most environment that you can generate in order to study route integration. And there's only a single turn along the route, and it's a 90-degree turn. So I'm not entirely sure that vestibular information or head direction information would help. But I mean, I don't want to rule it out.
Sure. Well, I guess maybe our next speaker, Paul Smith, will have also something to say about that, looking at the, say, the impact of vestibular lesions or vestibular dysfunction, even on, say, hippocampal adrenal processing. Maybe one more before we move to Paul. So Wayman Moore asked, "Great talk, Jan. So is that desktop VR without inertial motion cues? Is it that the lack of motion cues which makes older people difficult to finish the task?" So again, another question on motion cue.
It is a very similar case. Unfortunately, I can't answer this. It's even worse. I didn't even tell you that this was all done on a tablet. It wasn't even a computer screen or an immersive virtual environment. It was a simple small tablet that participants used in a real task. I mean, I'd love to run that experiment, but we haven't done it, so I can't really comment.
Okay, well, I can't find another question about self-motion, but then let's have at least one final question here from Mary Haggerty. What do you think would happen if you removed the landmarks for the landmark group and gave them more trials?
Isn't that essentially the identical landmark environment that we had, where the landmark was just non-informative?
Well, I suppose maybe about if you remove landmarks entirely, you know.
I believe the identical landmark environment is essentially the same as an environment without landmarks. So we just originally this was supposed to be an eye tracking experiment. So we wanted to have essentially placeholders in the identical landmark condition. But since the landmarks were all identical and if you approach the intersection from north, south, east, west, the intersection would look absolutely identical. It is as if there were no landmarks in the identical landmark environment.
Sure. Okay, well, I think we have to move on to Paul. So thanks so much, Jan. Have a good evening in Bournemouth. And we're now switching continents again and move to New Zealand to Paul Smith. Paul, can you hear me?
Yes, I can. Can you hear me?
Yes, we can. Fantastic.
Okay.
So yeah, Paul Smith from the University of Otago. I was going to talk about morphological changes in the hippocampus and entorhinal cortex as a function of age-related vestibular loss. So Paul, the stage is yours.
Thank you. Good morning, everyone, from all the way from New Zealand. As Thomas said, I'm going to talk about structural changes in the hippocampus and related areas in relation to vestibular loss. Many people don't realize just how common vestibular dysfunction is. In fact, up to 50% of all consultations with GPs are thought to be due to vestibular-related dizziness. And several years ago, a study was published from the United States by Yuri Agrawal and colleagues, reporting that about 35% of US adults actually suffer from vestibular disorders, and that statistic increases with age. In addition to the usual causes of vestibular damage, such as mechanical trauma and infection and tumours, the vestibular system also degenerates with age as part of the normal aging process. And over the last several years, there's been increasing evidence that noise trauma that causes hearing loss can also cause vestibular damage, in particular damage to the otolithic part of the vestibular system. So it's no surprise that vestibular impairment has been related to a large increase in the probability of falling, some 12-fold increase in falling, which of course is a major cause of death in elderly people. Finally, bilateral loss of vestibular function in some recent studies has been related to an increased odds or an increased risk of Alzheimer's disease. And that may be partly also related to the fact that hearing loss is related to an increased risk of Alzheimer's as well. So there's been a lot of work on the effects of vestibular lesions on the hippocampus over the last couple of decades. and I don't have time to go through any of it in detail, but because I want to focus on structural changes, but it's been shown that bilateral global vestibular lesions, and by global I mean complete loss of vestibular function of all the different parts of the vestibular system, cause disruption of hippocampal place cell function. Theta rhythm is disrupted, including in the entorhinal cortex. There's a disruption of LTP. There's an array of neurochemical changes involving a decrease in muscarinic acetylcholine receptors, an increase in NMDA receptors. There are changes in dendritic complexity in CA1 in rats. And finally, it's been shown that there are decreases in the volume of the hippocampus in humans. And the exact nature of these changes depends on the type of vestibular damage. But in the case of complete bilateral vestibular damage, it's been reported that there's a bilateral atrophy of the hippocampus. And the first study to report this was one that we were involved in with Thomas Brandt and Michael Strupp. And this was a study where 10 people who had bilateral vestibular loss for about 8 to 10 years showed an atrophy of the hippocampus bilaterally of about 17%. And this was selective to the hippocampus. I'm not sure if you can see my cursor on the screen, but the bilateral atrophy of the hippocampus in these patients was related to impairment of spatial memory as measured in a virtual Morris-Water maze. So this task is similar to the rat Morris-Water maze, except the patients were sitting in front of a computer screen, and they had to navigate to the correct quadrant, remembered quadrant. And what we found was that they spent less time, the bilateral vestibular lesion patients, in the correct quadrant, and also they showed a greater heading error. And these sorts of results have been reported by other people. So that was the first report of an atrophy of the hippocampus after complete bilateral vestibular loss. Now, within the vestibular system, many people will be aware that there are 3 semicircular canals that detect angular acceleration in different planes, but also 2 otolithic structures called the utricle and saccule, which get somewhat less attention generally. They're harder to get to. You can see on this slide here that down on the left-hand side, the utricle and saccule are most notable. And we spent some time developing a surgical approach in the rat to expose the utricle and saccule. And you can see this on the bottom right-hand side, the utricle on the top right, and the saccule beneath it. And we did this so we could electrically stimulate the utricle and saccule selectively, independently of the semicircular canals, in order to look at the transmission of information from the vestibular system to the hippocampus. Now, the otoliths are very interesting because, in evolutionary terms, they're very, very old. The oldest form of the otoliths actually goes back an estimated 670 million years. And a very primitive version of the otoliths exists in species like jellyfish, and they're called statoliths, and they allow the jellyfish to determine its orientation to gravity, because one of the things that the otoliths do is detect linear acceleration by gravity. So if we look at the evolution of the different parts of the vestibular system, the otoliths go back 670 million years and evolved long, long before the semicircular canals and before the cochlea, which suggests the possibility that otolithic information, given that it involved detection of gravitational vertical, may be a fundamental kind of information for the brain in developing coordinate systems for spatial navigation. And this shows a depiction of the orientation of the utricle and saccule in the human temporal bone. And what I want you to notice here is if you look at the saccule, because of the orientation of the saccular hair cells in the head, those hair cells are going to be in a position to detect linear acceleration by gravity. So when the head is tilted laterally, for example, left or right, those saccular hair cells are going to detect a change in the force of gravity and tell the brain that the orientation of the head has changed. And one way of measuring saccular function is through a reflex called the circular vestibular evoked myogenic potential, or CVEMP. And this can be activated with sound, for example, 500 hertz at greater than 100 dB. And it activates a fast reflex that goes from the saccule in the peripheral vestibular system down to the sternocleidomastoid muscle. And you can measure that contraction, and it gives you an index of cervical vent function. There's another one called ocular vent, which is a measure of utricular function. But I'm going to talk about the CVEMPs. So we can use the CVEMP as a measure of how good the saccula is functioning in humans and also in rats. We've been interested in the transmission of information from the vestibular system in the inner ear to the hippocampus for quite a long time. And we're interested in what routes, through what routes this information is transmitted, but we're also interested in how the hippocampus might use different kinds of vestibular information. For example, otolithic information about linear acceleration from the utricle and saccule, versus information about angular acceleration from the semicircular canals. So we set about selectively stimulating each of the structures in the inner ear, the horizontal canal, the anterior canal, the posterior canal, utricle, saccule, and cochlea in a series of studies, and then recorded local field potentials in the hippocampus using a 16 electrode microarray. And so what we're looking at here are heat maps showing the results of stimulation of the different structures on the hippocampus. And this field potential is usually triphasic, so we can divide it into phase 1, phase 2, and phase 3, going down the screen. And in each case, the first heat map shows amplitude and the second one latency. And I just draw your attention to the utricle and saccule over here. We also stimulated the cochlea for comparison, because we're very interested in dissociating the contributions of hearing to hippocampal function from vestibular responses, or the effects of vestibular stimulation on hippocampal function. What we found, the hot colors here represent the highest amplitude and longest latency. What we found was that when you stimulate the saccule and the utricle, you get quite strong responses from the same stimulation, 300 microamps. You get strong responses in the hippocampus, but particularly on the contralateral side to the stimulation. And if you analyze these results statistically, it turns out the utricle and saccule have the strongest responses compared to the semicircular canals, for example. So there's a lot of otolithic input that gets transmitted. And I want to emphasize here that the stimulation of these structures was highly selective. We monitored eye movement, and we used bipolar electrodes and did various controls to make sure the stimulation was selective. And if we just zero in on the saccule, here's an example of the effects of the same stimulation at 300 microamps in different parts of the hippocampus. And so these are the triphasic field potentials that you get from different areas of the hippocampus. And you can see that the potentials vary in amplitude and latency. So, this naturally leads to the question, what is the saccule giving to the hippocampus? What is it doing with that information? and how might that change under conditions of vestibular disorders or with aging? Now, Yuri Agrawal at Johns Hopkins, with whom I collaborate, has done a number of studies showing that the C-vent function, cervical vestibular evoked myogenic potential function, decreases with age. And here are two examples on the left and right. On the far left, this is latency, which doesn't change very much, but you can see on the right-hand side of that figure and also on the right-hand side of the slide in the other figure that C-vent amplitude goes down with age. So it's fairly clear that otolithic function, not just in terms of the saccule, but also the utricle decreases with age. Therefore, you might expect that with age, there would be some effect of reduced saccular function on areas of the brain related to spatial memory. So this is a study that we published just recently that was done at Johns Hopkins, and I was involved along with a lot of other people. And in this study, we used 80 subjects from the Baltimore longitudinal study of aging, and the mean age was 77.5 years. And these people had C-vents measured as a measure of saccular function, and at the same time they had MRI scans. And what we did is we used the MRI information to then develop a data set on the volume of different structures in the brain, including the hippocampus, entorhinal cortex, amygdala, thalamus, and striatum. And from there, we also developed 3D models in order to do a shape analysis of these different structures. And then we used multiple linear regression and logistic regression to see whether we could predict the changes in volume and shape of these structures from age, sex, intracranial volume, and C-vent amplitude. And what we found is that a reduced C-vent amplitude, indicative of reduced saccular function in these people, was statistically related to a statistically significant decrease in the volume of the hippocampus bilaterally. And also, intriguingly, it was also related to a decrease in the volume of the entorhinal cortex, but only on the left-hand side. So these results are somewhat consistent with other studies showing that complete vestibular loss is associated with hippocampal atrophy. Also, in terms of the shape analyses, and this is a bit of a complicated picture, and my laser pointer is not working here, unfortunately, but if you look down on the bottom right-hand side, you can see in blue some patches on the hippocampus, and the blue patches indicate that a lower C-vent amplitude was related to shape compression in the hippocampus, consistent with a decrease in volume. So the blue in this picture indicates compression of the shape, and red indicates that lower C-vent amplitude, reduced saccular function, was related to expansion of a shape. And so we found that poor C-vent function was related to shape compression in the hippocampus, entorhinal cortex on the left-hand side, and also the amygdala. Interestingly, we found shape expansions in some other areas like the striatum that I won't talk about just now.
Paul, 3 minutes, please.
Okay, thank you. So these results raise the question of the importance of otolithic function in the vestibular system to spatial navigation and spatially responsive neurons in the brain. And so the results are somewhat consistent with some previous studies in rodents and humans. For example, it's been shown that if you take a transgenic mouse strain like OTOP mice or het mice, who develop without any otoconia and therefore have no otolithic function, but have normal semicircular canal function, they exhibit spatial memory deficits in standard tasks like the radial arm maze. Also, human studies have shown a connection between poor C-vamp amplitude and impaired spatial memory in a number of studies now. Jeff Torby and Ryan Yoder have reported previously that mice without otolithic function exhibit an impairment of head direction cell function. And a couple of years ago, Ryan Yoder and company reported that these otolith deficient mice also have an impairment of place cell function. And lastly, it has been reported previously that complete vestibular loss is associated with a reduced hippocampal volume in humans. So putting it all together, it suggests that the otolithic part of the peripheral vestibular system may be particularly important in spatial memory systems, which is not to say that the semicircular canals are not, but the otolithic part of the vestibular system is the most ancient part of the inner ear, and it's also the part that encodes linear acceleration by gravity, which one would think might be particularly important for the brain because it is very old in evolutionary terms, and also because detecting which way is up is a pretty fundamental objective for survival. So lastly, this is just a diagram summarizing this idea that vestibular loss involving the otoliths, and maybe particularly the saccule, as a result of aging, and then a variety of other factors, even noise trauma, as well as mechanical trauma and tumors and infection and so forth, may cause hippocampal impairment and impairment of related structures, which then leads to cognitive problems that particularly obvious in relation to spatial memory, more than other kinds of cognitive functions. And there's a clinical relevance to this, and that is that there are patients who suffer from otolith function specifically, and to date, most of the artificial vestibular implants around the world have involved an implantation of electrodes into the semicircular canals only. And these people tend to suffer from some strange illusions, presumably because they're getting semicircular canal stimulation. These are people who would have bilateral vestibular loss and probably hearing loss as well. And they have electrodes implanted in the semicircular canals. And because they have no otolithic stimulation, the information about angular acceleration is being sent to the brain without any corresponding information about gravitational vertical, which is a problem. So surgeons have been working on the idea of stimulating the otoliths, and it's particularly difficult to do this, but a group in Spain has managed to do this, and I think also Charlie Della Centena at Johns Hopkins. And the reports so far are quite promising that with selective stimulation of the otoliths, you can achieve an improvement in posture. The reports at the moment are reasonably qualitative, but it looks like this might become a clinical reality in the not too distant future. So I just want to finish by thanking a very large number of collaborators, Yuri Agrawal, who led the MRI study I just talked about, and other people involved in that included Christoph Lopez, Tilak Ratnatha, Elliot Rebello, Susan Resnick, Daniel Tord, and Eric Way, and then other people who were involved in the hippocampal mapping studies in rats that I mentioned. Thanks very much.
Okay, great, Paul. Thanks so much. Very interesting talk. So let's have a look at the questions. So the first one would be from Arne Ekstrom, who's wondering if you could speculate on the causal direction of the vestibular effects on hippocampal volume. Could it be that these individuals are sedentary and thus less active, affecting the hippocampal volume?
That's certainly a possibility. There's no question that particularly people who have complete bilateral vestibular loss have limited activity. Many of them become housebound and may even suffer from agoraphobia. So it's very difficult in these human studies to dissociate the indirect effects of vestibular loss from the direct effects of vestibular loss. Interestingly, on that subject, in rats, it hasn't been found that bilateral vestibular loss causes hippocampal atrophy, which may be related to the fact that rats with bilateral vestibular loss are very hyperactive. Mm-hmm. However, we have found changes in dendritic trees, and in CA1 we found that they start to essentially decrease in complexity. So yeah, I couldn't discount that in humans.
Okay, cool. Second question from Laure. Does vestibular loss have an effect on cerebellum volume or cerebellar volume or function?
Well, as far as cerebellum volume goes, as far as I know, Adolfo, who's here, may have a comment on this, but as far as I know, no study has reported a decrease in volume, and also people have looked. In that study we did in 2005, the hippocampus was the only place that atrophied. As far as function goes, I predict widespread changes. There have been a lot of studies showing many electrophysiological, anatomical, and neurochemical changes in the cerebellum, and there have been recent studies, including one from Laura, I think, published recently, reporting a transcerebellar projection to the hippocampus. And so, one thing that's been fairly ignored, I suppose, over the last decade or so is the cerebellum, of course, gets direct input from the vestibular nerve into the flocculus. And so one potential route for information from the vestibular system to the hippocampus is via the cerebellum. And so I think that, probably there are many important functional changes there that may then affect the hippocampus.
Okay, cool. Next one from Tim McNamara. Is there any way to exercise the vestibular system? In the comments, one of our attendees even suggests headbanging to death metal. So is there a way to exercise the vestibular system?
That's a wonderful question. That is a wonderful question. There's a thing called galvanic vestibular stimulation, or GVS, where you put electrodes on the mastoids, and it's used around the world clinically as a diagnostic tool. If you put the cathode on the right side and the anode on the other one, what happens is the person will feel as if their head is being rotated toward the side of the cathode. Anyway, it turns out that this kind of stimulation, GVS, it's been applied in a lot of experimental areas to even at subthreshold amplitudes for the treatment of things like hemispatial neglect, Parkinsonian symptoms, neuropathic pain, and there's a theory that if you add a stochastic signal to it, so-called stochastic GVS, it might actually, by stimulating the vestibular system at a very low level, it may actually change EEG rhythms and so forth. We've actually just got a grant to study its effects in rats on the striatum, because not very much is known about how it actually affects the brain. But GVS itself will actually activate the vestibular system. And so I think there was a paper out just recently, a medical hypothesis suggesting that if you stimulated the vestibular system that way, maybe you could actually change metabolic activity by essentially telling the brain that you're moving when you aren't. But I don't think there's any evidence for that. Interesting idea there.
But is there then any work looking at the effects of GVS then maybe on hippocampal function or spatial cognition more broadly?
Well, actually, we did do a study some years ago looking at the effects of GVS on hippocampal neurogenesis. And strangely, what we found was that GVS in rats actually reduced hippocampal neurogenesis, actually reduced it. It didn't have any effect on spatial memory in these animals, though. Vestibular loss has been shown to modulate neurogenesis, and one of our ideas is that because the vestibular system is telling the brain when we're moving, that maybe vestibular stimulation is actually involved in the regulation of neurogenesis in the dentate gyrus, but that hasn't actually been explored in any systematic fashion. Again, a fascinating idea.
Okay. Good. Maybe one last question from Jean-Laurence. You see that poor vestibular function correlates with poor performance in spatial navigation tasks in VR when subjects are seated and therefore the vestibular system is not recruited. How does this relate with navigation during active locomotion?
Oh, I see. Okay. That's a difficult question. My understanding is that although studies have been done on spatial navigation and virtual reality, and people with poor vestibular function perform worse in that situation, there's no really good data establishing any connection between what happens with real navigation. I mean, of course, in that situation, what you're getting is you're getting visual stimulation, like optokinetic stimulation, which is going to activate neurons in the vestibular nucleus through subcortical pathways involved in the optokinetic reflexes. And so I would expect that if you've got poor vestibular function, I think it's been shown by Thomas Brandt and others that what tends to happen is there's a sort of tension between the vestibular areas of the cortex, like the parietal cortex and the visual cortex. And so what happens when you have poor vestibular function is the visual system becomes more dominant. But you would expect with poor vestibular function that it would affect the integration of visual information. And I would predict that one of the reasons for the poorer performance in virtual reality situations is because when you take the vestibular information away, the visual information becomes more ambiguous. However, it is a very different situation from actually moving. Yeah, that's a great question, and I don't have a great answer for it. I don't think anyone really knows, to be honest with you, but it's a very good point.
Right. Well, I think it might be the perfect transition to tomorrow, because tomorrow we have two more talks on vestibular dysfunction and navigation and on human work. So, yeah, thank you, Paul, very much. That was a great talk.
You're welcome.
I guess you can go back to sleep now. Right, so this concludes our session on aging and navigation. So I want to thank everyone for joining in, for contributing to the discussion. I saw in the chat window that some people said they could listen to another couple of hours of talks, or maybe that talk. Same here. It's a shame that we're already over, but the good news is tomorrow we're going to have 3 sessions, so you can actually enjoy many more talks than today. Yeah, I hope you enjoyed it. I hope you found the discussion stimulating, and we hope to see you all back tomorrow. And have a good day, good night, and bye-bye.